Hola, yo soy Laura Corín de Argentina.
Yo soy Guilherme Pedrini de Brasil.
Y yo soy Joaquín Díaz de Chile. Y esto es Fase 3, tu podcast de análisis crítico en linfoma y leucemia linfocítica crónica.
En español
y en portuñol.
Bienvenidos todos a la cuarta entrega de nuestro podcast Fase 3. Este es nuestro primer podcast con un entrevistado. Y wow, qué entrevistado. Pero primero voy a presentar a mis coanfitriones, Laura Corín.
Buenas tardes, un gusto estar acá con ustedes de nuevo.
Siempre tan formal, Dios mío. Y Guilherme Pedrini.
Hola a todos, hola Laura, hola Joaquín.
Bueno, en realidad este es como un podcast entrevista, pero comentario también, porque la entrevista ya está grabada, pero la entrevista es en inglés. Entonces, lo que vamos a hacer es un poquito dar el escenario clínico en que motivó esta entrevista y comentarios respecto a las opiniones del entrevistado, que han sido muy entretenidas. Entonces, todo esto comienza con una carta al editor en el New England Journal of Medicine de agosto 24, 2023, llamada Cell of Origin Subtypes and Therapeutic Benefit from Polatuzumab vedotin. Autor principal Alan Palmer de Chapel Hill, David Kurtz de Stanford y el senior author Ash Alizadeh, hemato-oncólogo de la Universidad de Stanford, que tuvo la amabilidad de venir a esta entrevista con nosotros. Esta carta al editor habla de, digamos, defiende a través de la observación en diferentes ensayos clínicos en escenarios recaídos refractarios, en los ensayos fase 1, fase 2 y en los estudios post-marketing, así como en el ensayo clínico Polarix, esta eficacia diferencial del Polatuzumab en diferentes subtipos de célula de origen, principalmente en el no centrogerminal, predominantemente célula B activada. Y esto, bueno, trajo muchos comentarios y muchas preguntas, y por suerte tuvimos la posibilidad de entrevistarlo y nos dio unas opiniones muy interesantes. Aquí lo tienen, Ash Alizadeh en fase 3. Thanks, Dr. Alizadeh, for coming to our podcast.
Thank you for the invitation.
I'm gonna go first, if it's okay with you. So you mentioned the differential efficacy in ABC subtype of Polatuzumab vedotin, and we recently had in ASH last year shown the remodel B study showing the same tendency with bortezomib, which is much cheaper, for example, for Latin American countries. So in ABC subtype, would you choose one over the other, and why? And if you consider that the meta-analysis published recently in Annals of Hematology showing benefit of Polatuzumab over the other agents in ABC subtype.
Great, thank you. So in the United States, bortezomib does not have an indication label for use in large B-cell lymphomas. So its use in large B-cell lymphomas is generally considered off-label, and even though it may be cheaper, the logistics of getting it approved are somewhat difficult. With that being said, having looked at the Remodel B study and the subsequent analyses, honestly, I was a little less impressed with the therapeutic benefit of bortezomib in that study. As you remember, the Remodel B study was focused on a pre-selected population expected to benefit from bortezomib, and the primary analysis did not identify that benefit. It was the subsequent unplanned analyses of that study that I think in part relied on excluding molecular high grade, if I remember correctly, as the basis for seeing the benefit, and doing that exclusion based on a tool that's not widely available to us, a molecular tool that the British group has developed that we don't have accessible to us. Now, I will admit that for Polarix, this is also an unplanned analysis. Well, it was a planned analysis in the clinical trial. It was just not a primary or secondary endpoint with dedicated power considerations. It was a planned correlative of the study with dedicated molecular profiling. And, you know, we were very excited to see these results, the overall trials being positive, but a little bit surprised by the breadth of the approval label in the United States across the entire landscape of large B-cell lymphoma and separately high-grade B-cell lymphomas, which we tend to usually treat with more intense regimens like infusional regimens like EPOC, or generally not using the R-CHOP itself backbone. So I don't know if I answered your question. Between these two regimens, only having one of them approved in the United States for frontline large cell Polatuzumab, I tend to favor using that. But I hear what you're saying about the costs of these drugs. I guess what I don't remember from the Remodel B analyses, did that also result in an overall survival advantage, or both EFS and OS, right?
Yeah.
So it's interesting. So do you use that in Latin America now, or thinking about it?
No, we can't use it because it's off-label. And recently we have Polatuzumab approved in Argentina in first line. We used to have it only in relapse refractory patients, not transplant candidates, but now we have it in first line also. And we have like a real conflict with that because the BFS benefit for the whole population is like really low. And the expense, the increase in the expense in the cost of the treatment is extraordinary. So, and patients read about the new treatments and want to get the new treatments, but if we use it in all the DLBCL patients newly diagnosed, the whole health care system will collapse. So we have to, your letter seems interesting because it gives us proof to select patients and not use it in all the patients without proven benefit.
Yes. Yeah, in fact, you know, the letter kind of came after an online discussion and some discussions here at Stanford around institutional practice and selective use of the drug. Of course, you know, an approval doesn't necessarily force us all to use the same regimen for every patient in every setting. There is the practice of medicine. But it seemed surprising to me when we said that we would be selective, to hear from so many colleagues to say, well, you know, 20-something countries have had approvals. NCCN is giving a Category 1 recommendation in the United States, and it's a broad approved label, 11-2 vote in the US by the Oncology Drug Advisory Committee to the FDA, that the burden of evidence that these folks needed to see is all past. Everybody should get Polatuzumab. And in all honesty, the way that I thought about it was, well, this is a drug that does improve PFS. It's dramatic that has taken us 2 decades to get here. But the magnitude of the effect, as you said, is small. You have to treat, I think, 16 patients for one to benefit in PFS, but not OS. And what is the number needed to harm? You know, thinking about 5 to 7% increased rate of hematologic side effects, including febrile neutropenia, especially in older patients where the rate of febrile neutropenia was 20%. If now you have 1 in 5 patients that you need to treat to potentially hospitalize somebody for febrile neutropenia, I thought we should be a little bit more guarded about the broad use. And honestly, the FDA voiced some of these same concerns in the US when they were during the ODAC panel listening to them to say, show us the uniformity of treatment. And that's sort of what motivated our search to say, was there a group that benefited? But of course, we didn't make this data, right? This data is all published. We just considered the data from all the publications and said, what do they show in relation to cell of origin? And it's pretty consistent. And we didn't pick these papers. They're the only papers that are available on the topic. I personally use this drug now mainly for ABC patients because I tend to think that I need Polatuzumab for a rainier day for the other patients in case they relapse, and I need it as a salvage or bridging agent in a rainier day than the PFS benefit that is the first line. So that's the way I've been thinking about it. And hopefully that thinking will be borne out by more analysis.
So if I'm getting this right, the hypothesis behind the letter is that the efficacy is diluted, pulling the GCB and the non-GCB subtypes with Polatuzumab vedotin. So does this mean we do not need a randomized control trial, or do we need a randomized control trial in this subgroup?
Well, ideally, we would do another trial to address this point. We put the colon cancer example as an example because that is an example where Amgen came forward and said, we see this difference, that the EGFR antibodies don't work as well in KRAS mutant colon cancers, and 2 subsequent trials went on to show that that's right, that this drug only works in the subset without the mutation, and that's the way we use the drug. I don't know if Roche and Genentech will do such a study because it seems like they have full approval already in the United States and much of the world. I suspect what will happen is folks will try to replicate these results in population studies, like the post-marketing studies. We looked at a few post-marketing studies in Israel and Taiwan that were available of Polatuzumab, and they showed the same effect. But can you show that the different groups are different and how much they benefit? Of course, it depends on how you measure cell of origin. You know, the studies we looked at used traditional immunohistochemistry or RNA-based methods, and it didn't seem to matter. They had the effect was larger for activated than or non-GCB than GCB. So it would be nice if there was another randomized study. I just don't know who would do it.
The same applies to the first line, because all the trials that were in the letter, most besides Polarix, were in the relapse refractory setting. So would that change anything, do you think?
The line of therapy, I think it could. Both the 365 trial where Rituxan and bendamustine were the backbone, and Polarix, so that was relapse, and the frontline trial, Polarix, where the backbone is anthracycline based, right? R-CHOP or RCHP, showed the same effect, the disproportionate effect, and when combining them, the dilution. So I guess I can't be 100% sure that other regimens could not erase this cell of origin specific effect. Like if you did Polatuzumab with CAR T cells, or Polatuzumab plus a bispecific, maybe the cell type specific effect would go away if that other agent is the equalizer. So I can't say for sure, but my sense is this is a pretty significant effect of Polatuzumab by cell of origin to neglect.
Do you have any biological rationale for Polatuzumab working better in the ABC subtypes? Like, is there more CD79B in the cells? Is it because of the mutations? Do you have any biological rationale for that?
Yes, so the original idea, as you know, for GCB and ABC with relation to these molecules was that the activated B cell type has chronic activation of the B cell receptor, and CD79B, as with CD79A, are one of the three components of that receptor. There's CD79A, CD79B, and the immunoglobulin. That's the B cell receptor. Now, the amount of these molecules between GCB and ABC is not dramatically different, marginally different, but the activity of the pathway is different. There's chronic activation of the pathways, very nicely shown by Lou Stout group over the years. And genetic tools have shown us that if you ablate the B cell receptor by cutting out CD79, either with CRISPR, CD79A, or short hairpin RNA, you're disproportionately lethal to the ABC subtype. Now, that's cutting the genes out, not using an antibody drug conjugate. So when you use an antibody drug conjugate, why should it be more toxic to an ABC than a GCB? And I think you can think of two ways. One is a B cell receptor dependent mechanism, where this same receptor that is on hyperdrive and feeding the pathway more, just if the thing binds, maybe receptor-based internalization and dependencies of the cell, that is more lethal, or a B cell receptor independent mechanism, maybe the free drug, the MMAE that falls off of Polatuzumab or gets cleaved and some cells release it in the environment. I think those are both possible. There was an interesting presentation, a poster at AACR last April by Lou Stout's group that suggested that free drug may be responsible. Because as you know, the free drug from an antibody drug conjugate has very different pharmacokinetics than, say, our cheap drug like vincristine, where you give the drug, you go up, you come down, and then it's gone, right? Within ADC, the free drug has a very long half-life because it's gradually coming off the antibody as the body cuts the drug off. So those are the two ideas. I think it needs future experiments to address the mechanism more.
And do you think determination of cell of origin through gene expression profile or immunohistochemistry, because in our clinical practice, we don't have availability of gene expression profiling, and we just do it by immunohistochemistry through Hans algorithm. So do you think it has an impact? Do you think we can accept the error, the margin of error in cell of origin determination to choose Polatuzumab?
Yes, so that's a good question. As I mentioned, even here at Stanford, where cell of origin was originally described, we still don't use RNA for cell of origin determination. What's available clinically is immunohistochemistry and the Hans method. And I guess in that context, we have to take into consideration that 15% or so of patients who are considered non-GCB don't actually have ABC tumors. So are they not benefiting from Polatuzumab and are getting pulled in for no reason? I don't know for sure. I think within Polarix, you could probably address that. I know several folks who've tried to get access to Polarix unsuccessfully, case level data. But if somebody's willing to do that analysis by Hans, it would be nice. Now, there's some things about Polarix that are a little bit disappointing in that this large trial of 800 some odd patients did not have central pathology review. There was central review of circulating tumor DNA, RNA for nanostring of fish, but no central pathology review, which seems crazy to me. So there could be some differences across the world. You know, there were so many sites in cell of origin determination, especially as informed by the prior trials, like both Remodel B and Robust from Celgene, where they showed that there was some heterogeneity in cell of origin classification at centers. So that may make things harder. But in the real world, immunohistochemistry has to work despite the vagaries, right? If you're going to do patient selection the same way as we determine breast cancer hormone receptor status, we have to be able to determine cell of origin status. So we have these things better work with simple diagnostics like Hans in the average hematopathology laboratory without very complicated and expensive diagnostic assays. Even though I work on those assays, they need to work for every patient in every setting. So, but yeah, I better work with Hans, Laura.
You were the first author in one of the most important cell of origin papers that came back 23 years ago, and you've seen, you know, the whole story develop in a lot of negative trials, and now we're seeing the revival of the cell of origin, mainly with the Remodel, with the Pola. How does it feel to, you know, after all the negative trials, finally see this data coming out as a positive and probably practice changing?
Yes, so I think it is a little bit bittersweet, you know, having seen this difference between these subtypes and part of the work I did for my PhD. This is a big biological difference that we observed, but we didn't know whether it would be therapeutically tractable. And of course, a lot of people have tried ideas to try to overcome the prognostic difference between these two groups because there's such a strong molecular difference and such a significant clinical difference. And now we know genetic difference and epigenetic difference and so many other things. That's been easier said than done. And many drugs have been casualties of the drug development process over the last 2 decades. Lenalidomide, bortezomib, et cetera, have not really been able to, ibrutinib, in unselected populations showing the benefits. So I guess it is great to see that this may actually be overcomable in part with Polatuzumab. But I'm also super excited about, you know, cell of origin agnostic ways to improve outcomes, you know, with our T-cell focused therapies like chimeric antigen receptor T-cells and T-cell engaging antibodies, not seeming to have dramatically different efficacy by cell of origin subtype, and not even for double hit, it seems, for some of these things. So I think I'm excited to move the needle a little for patients. It just seems a bit frustrating that it's taken us so long to add an agent to the front line, despite having had some molecular insights, and I hope we have many more in the years to come.
What's your take on patients with high IPI score? Would you give them Pola-R-CHP, or would you stay with R-CHOP if they have a germinal center subtype?
With high IPI, well, germinal center subtype, we have not been giving Polatuzumab to, especially double hit, because I suspect those patients will actually might not only not benefit, but we may harm the patients if they really have a very high proliferative rate. If you look within Polarics, even among the high-grade subset, the median DTI, diagnosis to treatment interval, was pretty long, 27 days, which is substantially longer than our average patient with a high-grade lymphoma in an academic center, where we often admit them directly from clinic because they're so sick or so proliferative tumors. So we are not using this regimen for such cases. For high IPI, I think ABC high IPI we definitely would consider. I guess one challenge is that the way that the American system works is getting prior authorization for a drug like this in an outpatient versus inpatient is somewhat different. So if you wanted to use Polatuzumab as an inpatient, you would have to go through a different process, and many hospitals probably don't have Polatuzumab on their formularies, as opposed to the infusion centers where the patients are outpatients. So you have to go through additional hurdles to get Polatuzumab for an inpatient than an outpatient. But American logistics probably not as relevant for Latin America, probably.
We were going to see a lot of new agents coming on for these ABC subtypes. You know, there's the acalabrutinib trial going on. I believe there's some data for Zano, and also for new cell mod drugs. How do you think you're going to balance that, you know, regarding this patient with the ABC subtype? Do you think we're going to probably add on the polar chip backbone, or it's just going to compete with Polatuzumab in this scenario?
It's interesting that you bring that up. We were just at the international workshop for non-Hodgkin's lymphoma in Miami. Was it last weekend or 2 weekends ago? Last weekend. And Andrew Davies, who has one of those acalabrutinib studies that you mentioned, brought up that challenge for the Remodel A trial in the UK, where the approval and the NICE recommendation to cover Polatuzumab in UK has essentially jeopardized that study, the comparator arm. I don't know how it affects. We heard also from Simon Rule at AstraZeneca that the Escalate trial that has acalabrutinib has not been as hampered by the approval of Polatuzumab because of the other European countries where I guess Polatuzumab is not yet approved, or late in the negotiation process between the sponsor and the public health systems. But you're right, it does complicate things for those trials. What we heard that Laurie Sen from British Columbia presented, and her takeaway and thought was that R-CHOP should remain an acceptable alternative regimen for every patient, given the lack of a survival advantage, including patients on clinical trials. But your point is a good one, because if you're a patient and you see a PFS benefit and an approval and a level 1 recommendation, and now you go to an academic center or even a non-academic center that has a trial available, and you're saying, I want to give you the old regimen, or it does put the provider and the patient in an awkward position. So I don't know if I answered your question, but I feel like R-CHOP, I agree with Laurie Sen that R-CHOP should stay an acceptable alternative, not just for the health economics, but for the overall benefit in the population. I guess, could we say that ABC patients should also get R-CHOP if there is indeed a survival advantage? I don't know for sure. I don't know.
Thank you.
Okay, thanks, Dr. Alizadeh. I think we are very happy that you have been in this interview. And just before we let you go, are we there with ctDNA for MRD measuring in the LBCL? Is the evidence there yet?
I think the evidence is starting to become more concrete. I think we are still substantially behind our sister diseases for lymphoma for MRD, like CML as the first, ALL as the second, myeloma as the third, and CLL maybe as the fourth. I think lymphoma is probably the fifth disease where MRD emerges. And I guess AML, I forgot, I'm sorry, as kind of a key thing, but there are lots of steps still to take to use this as a biomarker for response assessment. But for drug approvals, we still have a little ways to go. And a lot of people are working very hard toward qualifying such a biomarker for drug approval. There will be a fair amount at ASH this year that I encourage you to see around MRD and lymphoma with ctDNA, in working the body of evidence that agencies like the FDA and others will need to see to consider approving drugs based on MRD.
Okay. Thank you very much for being here.
Of course, of course. Take care.
Thank you.
Goodbye.
Really
Bye-bye.
Pleasure.
Bueno, bueno.
Really nice guy.
Bueno.
Wow.
Laura, ¿quieres partir tú con algún comentario respecto a lo que hablamos?
A ver, creo que por la aplicación que tiene en Latinoamérica me parece muy importante lo que charlamos de la determinación de la célula de origen. Nosotros leemos todos los ensayos clínicos en los que la determinación está por el perfil de expresión génica, eso es algo que no tenemos accesible en la práctica clínica, y el mismo Ash Alizadeh dice que ahí en Stanford, él, que es el creador de la célula de origen, sigue utilizando el algoritmo de Hans por inmunohistoquímica como método de determinación de la célula de origen, y esto es importante para aplicar estos resultados que charlamos de esta letter al New England. Así que en parte destaco un poco eso porque es parte de nuestra realidad, la dificultad que tenemos en los métodos diagnósticos.
Me recordó esa frase de un podcast anterior que dijiste tú de que nos estábamos como cayendo del mundo o algo similar, y como que me volvió la esperanza después de escuchar eso, ¿no? De que en Stanford
Sí.
ocupan el algoritmo de Hans y no RNA Genomic Profiling o algo así.
Interesante la discusión de célula de origen, pero también su opinión acerca de R-CHOP permanecer siendo una rama controle aceptable para nuestros pacientes. Creo que esto es una discusión que vamos a tener muchas veces en los próximos años, principalmente si Pola R-CHP se va a tornar un standard of care en nuestra región. Pero creo que es una discusión que es muy compleja, y me quedo más tranquilo de saber que los grandes nombres de linfoma concordan que podemos continuar con R-CHOP.
Y lo mencionó
creo,
directamente, ¿no? Laurie Sen, Simon Rule, dijo, todos ellos de alguna manera opinan que en esta aprobación que es amplia, sigue siendo éticamente una opción válida el R-CHOP.
sí. Creo que el estudio, el análisis de esta letter es muy elegante, porque hay un análisis de todos los estudios, una análisis agrupada de estos estudios que demuestran claramente que el beneficio del Polatuzumab es muy mayor en los pacientes con células activadas. Y creo que eso es importante para nuestra región, que vamos a tener un impacto financiero muy importante de Polar-CHP. Entonces, hoy estoy más convicto que só voy a considerar Polar-CHP para pacientes con células B activadas. Pero creo que essa é uma discussão que temos que ter individualmente em cada centro. Pero para mim, esta letter é practice changing. Creo que devemos cada vez mais ter estes dados para que possamos tomar nossas decisões clínicas.
Como que quería destacar otro aspecto, principalmente en Latinoamérica, que no tenemos acceso a drogas nuevas para la segunda línea, las líneas sucesivas de tratamiento, que nuestra única opción actualmente es un ensayo clínico que no está disponible claramente en forma masiva para los pacientes, o el Polatuzumab en la mayoría de nuestros países. Aquí tiene CAR-T, pero no es la realidad de Latinoamérica en su globalidad. Y él mismo decía que le parecía importante reservar, aún en Estados Unidos, que tienen acceso a todas las drogas nuevas, reservar el Polatuzumab en algunos pacientes para esos días más oscuros, o sea, para los momentos, para pacientes que recaían. Y me parece todavía que eso es un concepto aún más importante para nosotros en Latinoamérica que allá. O sea, que habiendo disponibilidad de todos los agentes, que él considere que algunos pacientes habría que reservarlo, o sea, que no es necesario la primera línea, sino reservarlo para las líneas sucesivas, me parece muy importante.
Digamos, consistente con tu opinión respecto a estas moléculas, que has dicho siempre que, bueno, tenemos que en Latinoamérica encontrarlo, anicharlas bien, y él dio como una respuesta bien inteligente relacionada con esto. Oye, tenemos, digamos, sobrevida global al final demostrado, esa es una indicación. ¿Dónde estaría mejor la indicación aquí? Probablemente también hay que buscarla. Y creo que para mí me gustó mucho su reflexión respecto al ensayo clínico Polarix, de que finalmente este desenlace que tiene una magnitud de efecto bien pequeña, bien marginal, uno podría decirlo, probablemente tiene cierto grado de dilución en este contexto, yendo a buscar, digamos, el laboratorio, quien los culpa, una indicación más amplia, ¿ya? Y que evidentemente nos dejan al resto aquí tratando de sacar, digamos, las cuentas específicas de cómo vamos a interpretar eso. Y chuta, esto nos va a dar mucha conversación por un tiempo largo, creo yo, hasta que no tengamos acceso a otras cosas. Lo mismo que tu pregunta aquí relacionada con cómo sumamos esto a otros ensayos clínicos que probablemente tengan, digamos, una indicación también relacionada o determinada por célula de origen.
Sí. Es muy interesante que yo he hecho una pregunta acerca de esto, porque Ash Alizadeh fue el primer autor del paper que demostró la diferencia entre GCB y ABC. Creo que debe ser muy interesante ver cómo las cosas van cambiando. Y ahora, después de 20 años, después de muchos estudios negativos, nuevamente tenemos datos que demuestran que esta diferencia es muy importante. Entonces, creo que estamos, en mi opinión, estamos viendo un revival, un renacimiento de célula de origen en formas difusas.
Y es muy interesante que él lo defiende muy científicamente. No es solamente, oye, tuvimos una interacción en un subgrupo en un ensayo clínico. No, hay varias evidencias en ensayos clínicos recaídos refractarios, se suman con la de en primera línea, hay un mecanismo de acción potencial relacionado con esto. Eh, de alguna manera hay una señal bien clara y que lo que hay que determinar es, bueno, más precisamente si eso cambia manejo clínico o no. Y lo deja muy abierto, ¿no? Vamos a tener que seguir estudiando esto. Pero me pareció, pero on point sus comentarios. Muy bien, no nos queda más que dejarlos con esta entrevista a Ash Alizadeh, que le agradecemos de sobremanera este encuentro, esta entrevista, y hasta la próxima.